Welcome to today's Explainer. We're heading straight back to 1996, right into the middle of a dramatic, real-life medical mystery that played out in the southwestern region of Argentina. We are going to look really closely at a scientific investigation that absolutely shifted our fundamental understanding of virology. I want you to imagine a deadly pathogen suddenly deciding to just rewrite its own biological rulebook, literally forcing doctors and scientists to race against the clock to figure out how it was spreading. Today, we're going to follow the exact molecular clues those researchers used to finally crack the case.
Okay, let's dive into this. What exactly happens when a deadly pathogen breaks its own established rules? In epidemiology, there are certain laws we completely rely on to track and contain outbreaks. We expect a virus to have a known vector, right? A predictable method of transmission. But in the spring of 1996, doctors down in Argentina were staring down a terrifying scenario. This virus was behaving in a way that scientific consensus said was, well, simply impossible. It's a scenario that completely upended our understanding of how this specific pathogen operated.
To truly grasp why this was so shocking, we kind of have to look at the status quo of hantaviruses. Take the sin nombre virus in the United States, for example. The rock-solid, established rule was that hantaviruses are strictly transmitted from rodents to humans. Usually this happens out in rural areas, someone accidentally inhales contaminated rodent dust, and boom, they're infected. But critically, a human being is considered a dead end for the virus. You might get terribly sick, but you are not going to pass it to your family, and you're not going to pass it to your doctor. But the 1996 Andes virus outbreak, it shattered that assumption entirely. All of a sudden, hospital workers, spouses, and urban doctors who had literally never set foot in a rural area were falling ill with Hantavirus Pulmonary Syndrome. Let's trace how this tragic epidemiological web unfolded, because it happened fast, terrifyingly fast. It kicked off in September 1996 with what we call an index case in the small town of El Bolsón. When patients were transferred to a hospital in Bariloche, the unthinkable happened. Doctors and receptionists, people with absolutely zero rodent exposure, began getting sick. And the situation grew even more alarming when a healthy individual took a 20-hour car ride with an infected passenger and, sure enough, contracted the disease. Finally, the outbreak hit the massive urban center of Buenos Aires, striking a doctor who was simply caring for an infected friend. All in all, 16 people were ultimately caught in this deeply interconnected web of infection. So, the crucial point is the distance. 1,400 kilometers. That is the massive geographic gap between that initial outbreak in El Bolsón and the later cases popping up in Buenos Aires. Why does this number matter so much? Because it effectively, completely eliminated the standard theory of a localized environmental source. I mean, you can't blame a single localized population of infected rodents when your human victims are getting sick over 800 miles away from the epicenter, especially when those victims had never even traveled to the rural south. Because of this incredible spread, the standard rodent hypothesis completely fell apart. Investigators obviously did the logical thing first. They tried to find infected rodents near the victims, but the rodent trapping success rates near El Bolsón were surprisingly low, and here is the kicker. Absolutely no rodents were captured inside the actual homes of the patients. Not a single one. Furthermore, infected rodents they did catch, about 15 kilometers away, carried a completely different genetic sequence of the virus. So the researchers were forced to look for a different and honestly much more chilling explanation for how this thing was spreading. To solve this, the investigators had to turn to the very building blocks of the virus itself, using a process called viral sequencing. They essentially extracted viral RNA from the tissues and blood of the victims, and then used a technique known as RT-PCR to amplify specific parts of the virus's genetic code. Specifically, they were looking at the S and M segments of the viral RNA. You can think of this as a highly advanced molecular magnifying glass. By comparing the exact sequences of the virus found in different patients, scientists could see once and for all if they were infected by different rodents carrying varied strains, or, and this was the scary part, if they were passing the exact same virus among themselves. And this brilliantly illustrates what they found. The phylogenetic data was just incredibly clear. When you look at the viral family tree, there's a cluster labeled epilink/96, and there was a tightly grouped match. Every single one of those 16 epidemiologically linked patients, the people who shared hospital rooms, shared households, or were on that 20-hour car ride, they clustered perfectly together. They didn't just have similar viruses, they had the exact identical viral lineage.
Let's move to and see how this builds the case even further. This alignment created a literal genetic fingerprint. When researchers lined up those specific genetic codes, we're talking about looking at a total of 1075 nucleotides across those S and M segments. They found a 100% exact match among the 16 linked cases. The sequence, epilhung/96, shows the exact amino acid structure. It is essentially the molecular smoking gun, proving, beyond a shadow of a doubt, that the pathogen traveling from El Bolsón all the way to Buenos Aires was the exact same organism.
This comparison right here clinched the whole investigation. On one hand, you have the 16 linked patients sharing a 100% exact genetic match of the epilink/96 variant. But on the other hand, scientists tested unlinked local cases. These were patients identified as G and T who lived in the exact same town of El Bolsón at the exact same time, but they had absolutely no contact with the 16 linked individuals. While patients G and T had entirely different viral sequences. Think about it. If the outbreak was just caused by a massive rodent infestation, everyone would have varying local strains. The fact that only the people in direct close contact shared the identical strain was undeniable proof of the transmission method. So let's tally this up, because the mounting evidence was now undeniable. First, we have 16 epidemiologically linked patients sharing the exact same viral genetic code. Second, there were absolutely 0 unlinked hantavirus cases in Buenos Aires or Bariloche during this period. That means the virus wasn't just hovering in the local environments. Third, the infected rodents they trapped nearby had different viral sequences. And finally, fourth, every single one of the 16 patients had crystal clear paths of close interpersonal contact. Whether it was sharing a household, treating a patient in a hospital, or traveling together, the links were there. Which brings us to the truly monumental conclusion of this entire investigation. By brilliantly combining classical, shoe leather epidemiology with advanced molecular sequencing, the scientists had proven what was previously thought to be totally impossible. This 1996 outbreak provided the world's very first direct genetic proof of person-to-person transmission of a hantavirus. The Andes virus wasn't relying on mice to spread anymore. No, it was actively jumping directly from human to human. Now, what's really interesting about this slide is reflecting on what we used to believe. Researchers had previously assumed and confidently stated that human infection with hantaviruses was an accidental dead-end event, which typically is irrelevant for the maintenance of the virus in nature. This discovery in Argentina completely, entirely shattered that long-held safety net. It proved that a virus can mutate or possess unique biological properties that suddenly allow it to bypass its natural reservoir and use humanity as its new vehicle for transmission. It completely changed how we look at biosafety protocols, especially in hospitals dealing with sudden, unexplained respiratory outbreaks. Which leaves us with a rather chilling final thought for this explainer. The Andes virus showed us that nature is endlessly adaptable. And pathogens, well, they don't always follow the rules we write for them in our textbooks. If a virus previously known only to be a rodent-borne threat can suddenly learn to jump from person to person over a distance of 1,400 kilometers, what other biological rules are waiting to be broken in the natural world? It's a pretty powerful reminder of exactly why constant vigilance and molecular tracking are absolutely vital to our survival. Thank you so much for joining me today, and keep questioning the rules.